Aloha, my name is Amber Boettiger and today I will be presenting my senior capstone project on viral morphological analysis using quantitative transmission electron microscopy. Environmental virology is an important field as viruses are some of the most abundant microorganisms on the planet. They influence ecological, biochemical, and evolutionary processes in the ocean through their abilities to induce mortality of other microorganisms, alter nutrient and organic matter, and change evolutionary tracks through horizontal gene transfer. Each mouthful of seawater that you swallow contains around 10 million viruses, but these don't infect people or animals, instead bacteria and archaea. We don't know a lot about them because they are difficult to study as they lack the 16S rRNA gene used to track other microbes in the environment. Therefore, estimating their diversity across spatial scales relies mostly on metagenomics to compare their large viral assemblies of different environments. Metagenomics does not let us quantify the size and population of these communities, rather only demonstrates what was present at the time of sampling. Understanding the morphological features of these microorganisms can be a valuable tool to understand the ecology and diversity of aquatic viruses and their early stages of evolution and present day. In order to reach our sample, you must take a cruise ship about 100 miles off the Oregon coast and a submarine a few hundred meters down to the Juan de Fuca ridge flank. The viral communities here have not been explored, making this research push the known geographical limits of the virosphere deep into the oceanic basement and point to a wealth of novel viral diversity. This novel research could shed light on the early evolution of viruses, as Juan de Fuca exhibits a microbial community of deep subsurface geothermally heated fluid samples that mimic early life conditions. This fluid is accessible by boreholes penetrating deep into the sediment. There, the water is flushed onto tangential flow filters to be processed when back on land. The collected viruses are flushed off the TFF filter where they can then be added into a cesium chloride gradient. Viruses hold a unique property where they can assemble themselves across a gradient based on their densities. 3 cesium chloride densities, beginning with the heaviest density, are layered into a test tube. Then a viral concentrate is injected into the middle gradient and spun in an ultracentrifuge for approximately 3 days. After removal, distinct lines demonstrate the viral fractions that can be separated into their own sample set. 4 fractions were created to demonstrate the transition from lighter archaea viruses, sections A and B denoted in blue, into the heavier bacterial viruses, 1.38 grams per milliliter and heavier, denoted by C and D in red. These fractions can then be analyzed using their distinct morphologies in quantitative transmission electron microscopy, or qTEM. The study follows the methods of Brum et al. 2013, who completed qTEM on sea surface samples from Terra Oceans. In her analysis of over 1,400 viruses, she demonstrated 4 bacterial virus types, 3 of them being Claudio virillae, or tailed viruses, shown in red. My morphological analysis includes an additional 6 archaeal morphotypes, denoted in blue. All morphotypes shown except for tailed lemons, B, and filamentous envelope virons, G, were found in my analysis. Quantitative transmission electron microscopy entails spinning each of the binned viral fractions onto a glow discharge grid. These grids are then taken to the microscope, where photos are taken of the first 150 viruses from each grid that holds its own fraction. The 150 viruses are divided into 3 replicates of 50 counts to account for statistical significance in later analysis. After photoanalysis using ImageJ, each population demonstrated unique counts across each viral fraction. Population percentages from each morphotype are demonstrated from the lowest archaeal fraction, A, to the densest bacterial fraction, D. The bacterial viruses are shown in red and archaeal in blue to demonstrate the morphological makeup across the gradient. There is an evident shift from blue in A and B to red in C and D across the density line, supporting the assumption of changing community structures. While community changes are evident from the population percentage graphs, a Fisher exact test was used to determine if there was a significant difference between each of the fractions. The results demonstrated significance across all fronts except between the 2 archaeal fractions. The bacterial fractions remain statistically different, as D, or fraction 2.4, is the densest fraction, catching many broken viral pieces and altering the community structure. This is the first reported separation of archaeal and bacterial viruses using a cesium chloride buoyant density gradient. We accept the null of the existing gradient and with significantly different density fractions, further analysis could be completed on each fraction, such as capsid diameter and tail length. The results also open the door for increased investigations into archaeal viral ecology and virology. Thank you so much for listening, and I hope this presentation has given you a greater insight and appreciation for our ocean's smallest inhabitants, viruses.